In this video, we will discuss about some of the ways by which intra-tumor microbiota can affect the tumorigenesis, or it can promote the pro-tumorigenic effect and suppress the immune response. So we will understand different mechanisms that are possible by which intra-tumor microbiota helps the tumor cells to grow. First one is about this induced DNA damage. So in this case, what people have find out, or people have come to a point that this microbiota that is present within the tumors can release metabolites that will affect the DNA damage. So DNA damage is not only done by the change in the replication rate or through the uncontrolled growth rate, but it can also be done by the metabolites that are released by bacteria. For example, the E. coli that release PSK, E. coli that has this PSK uh, proteins that is released by E. coli can help in the DNA damage. So, and in the case of many cancers, the E. coli has also been found in the intratumoral microbiota. So that can also help in this DNA damage of the tumor cells. The other way that people have speculated or come to a point that is about this activate oncogenic pathways. So oncogenic pathways are activated in the tumors not only because of the tumor microenvironment, stress, or the change in the mutations, change in the DNA replication rate within the tumor cells, or some of the epigenetic changes that occurs in the tumor cells. But it can also be contributed by the bacteria. And one of the examples that they have uh, share about the bacteria that can cleave the E-catherine signals, thereby releasing this beta catenin to go into the nucleus and activate this NF-kappa beta pathway and activate this WNT signalings that results in activation of this NF-kappa beta pathway, which helps in the survival. The other ways is that the bacteria will release some of the cytokines that can interact with these TLR receptors, and those TLR receptors can activate these NF-kappa beta signaling pathways or P13K survival pathways that helps in the survival of the tumors. And the one more way that is shown here is through these cytokines that interacts with these receptors, uh, and these receptors can also trigger the survivals within the tumor cells, or can trigger the oncogenic pathways that helps in uh, metastasis or growth of the tumors. These things are not present in the detail here, but you can refer to this paper to go to the main papers where they have discussed about these details. Here we will only cover the different ways by which intratumor microbiota can act. The third one is this influence the immune host system. So in this case, in short term, we can say that the microbiota can also helps in immune suppressions, or we can say that it can cause inflammations, or that will activate the immune response. So it will release the cytokines, and one of the ways by which I will talk about the immune suppressions. First, let us uh, talk about the inflammations or promote immune response. So it will release cytokines where it will upregulate the cells, that is Th17, Th2, neutrophils, tumor associated macrophages, MDSC, myeloid derived stem cells, and it will enhance the immune response that will cause inflammations within the patients that helps in the tumor growth. The other way that it can work is through suppressions of the activated immune cells, and one of the way is through this regular PD-L1. In general, in the immune checkpoint case, what happens is that this PD-L1 that is expressed on the tumors interact with the PD-L1 receptors that are expressed on the immune cells, thereby suppressing the immune response. However, in this case, what researchers has found out in terms of the bacteria, that for example this E. coli bacteria have a FAP2 ligand that we have discussed a couple of times in our previous videos. So these FAP proteins can interact with the immune checkpoint on the tumor cell, on the immune cells, thereby suppressing the immune response. And there is this suppression of NK cells has been shown here, that the NK cells, B cells, and not just NK cells, yeah, NK cells, B cells, and T cells can interact with the proteins that is released by bacteria, that is here M. pylori or CAM1, that will cause the suppressions of the immune systems or immune response, we can say that. Other one is about the regulate anti-tumor drug resistance. So in this case, the drug resistance, in order to increase the drug resistance, the bacteria will release the cytokines such as DS regulatory cells that will leads to the suppressions of the immune system. Similar to the mechanisms that is shown in the panel C. So this is in this case what happens is that it will not suppress the immune system response. So instead of targeting the immune system, in the panel D case, there is cytokines will suppress the drug response, or prevent the drugs to reach to the tumor sites by releasing the cytokines such as Drex that will neutralize or deactivate this drug before it reaches to the tumor. So this is how it will regulate the anti-tumor drug resistance. Bacteria helps in the anti-tumor drug resistance. This is some of the ways by which the intratumoral microbiota mechanisms can work towards this tumorigenesis. You need to understand more about their mechanisms in detail, and with some examples you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.